Within the last few years, many companies and academic labs have shifted their efforts to applying AI methods to biology, whether it's building foundation models, virtual cells, agentic scientists, or just using ChatGPT as a therapist when your experiments fail.
In this episode, we talk about the promise and the perils of AI. We dive into a particular case where AI was used to predict enzyme function, and it gave results that looked really promising to editors, reviewers, and really anyone other than Valerie de Crécy-Lagarde, a scientist who's been working for over a decade on the exact problem that the AI paper solved. We were also joined by Rachel Thomas, co-founder of Fast AI and all-around AI expert, who gives us the broader view of how we can all make AI more useful in biology. A few years ago in 2023, there was a paper in Nature Communications where they used deep learning to try to predict the function of hundreds of enzymes, enzymes that were previously not annotated. They tested 3 of those in the lab. They confirmed the system worked. It seemed like pretty great application of how AI can be useful. But then, Valerie, you came across the paper. Tell us what happened.
Well, I've been working on trying to figure out the function of unknowns for like 20 years. And one of the 3 proteins they actually validated experimentally was one I had been hunting for the function for about the same time, like 15 years. This one was called YCIO, and it was a paralog, meaning a homolog with a different function of another enzyme I had discovered called TSAC. And TSAC is a very conserved protein. It's conserved from E. coli to human, and it's involved in a tRNA modification. And this is the field I work in. And this gene had been missing for 20 years. And we discovered the gene like nearly 20 years ago now. And so the papers we published were like 2009 and 11. We already showed in these papers that this paralog, YCIO, that is present in about 30% of the genomes, where TSAC, the 2, is present in every genome. So, and the similarity is very high. When you do a blast, you get both, and they're tough to separate. So immediately we said, is this doing the same thing or not? And we published that clearly it was not doing the same thing, but we didn't know what it was doing. And I've been giving this protein in my classes because I have a class where I give grad students unknowns to figure out. And YCIO was there every year for the last 15 years, okay? So imagine I see a paper, because they had YCIO in the abstract. Otherwise, I don't think I would have found this paper. So because they had YCIO in the abstract, it came up in my Google whatever feeds, and I was so excited. I was like, finally, they found the function of YCIO and this cool AI. And so immediately I read the paper, and immediately I saw that they fell in the same mistake that every grad student that had this protein to do fell. They said it is the same function as the TSAC, okay? And the EC number they give is the TSAC EC number. And the activity they test in vitro is the TSAC activity, except when you look at the kinetic parameters, they're about 4 orders of magnitude lower. So in vitro, when you take paralogs, they came from the same ancestor. They still have some of the capacity to do the ancestral activity. And in vitro, when we do annotation, in vitro alone is never enough, because you have a lot of things you could happen in the tube when you throw tons of substrate. And it's not the endogenous biological function. So the first thing I did, I was very low-key. I tried to do, I had never done this before, this matter arising they have in Nature Com is kind of a rebuttal thing. So I went, and you have to email the authors. So I wrote a little 2-pager just on YCIO saying, well, if you looked at the context and the literature and all that, you would have figured out that it could not be the function. So just beware when you do AI prediction to integrate more. And we put that in bioRxiv, but it was really just on YCIO. The authors didn't reply. So the guidelines are, if the author don't reply, you can send it to the editor. So I did that. That was like December 2023. So I did that. And the editor then asked the author that replied, and they replied totally out of scope. They were like, I don't even want to say what their reply was. So then the editor said, well, this is such a specialist issue. We're not going to publish your rebuttal. I'm like, what? Like, I'm showing that this is not correct and you're not publishing it? So I went mad. I went like, ooh, you know. So I'm like, okay, I'm going to do a paper, but I'm now going to go through the 450 predictions, okay, which I hadn't done at the beginning because it was a lot of work. And I won't go through the whole process because we went to the G3 genetics genomics because I had a friend there who's the editor-in-chief, and she says, yeah, we want the story, it's going to be really cool. And then they didn't have a rebuttal format, and she couldn't be involved because we were in conflict of interest. And the editors said to me, well, we don't have a rebuttal format in this format. So I had to rewrite the whole very thorough, very scholarly paper. And it took forever because the editors were never happy with what I was doing, but we got it. And I think the good thing is they made me really go through all the 453 predictions. And then I collaborate with Raquel Diaz, that Rochelle knows, who's my AI colleague, and she's just next door to me in the office, okay? So we really have a project together trying to do this correctly. So I got her involved, and that's how we kind of included XAI to try and understand why they were doing these errors, what was the source. So because I could classify the type of errors for her, then she could use XAI to kind of, so that's how it all happened, pretty much.
Wow. It seems kind of odd to me, like that a journal would not want to correct the record and instead, hope no one notices, or like what is, I'm not sure what the end goal is there.
I don't know. I don't know why I think he didn't realize how bad the paper was. So clearly it's a failure of reviewers. Reviewers should have caught this. So anyone who, there's one paper that a week that publishes a machine learning method to predict EC numbers. They're pretty much computer ways to give the enzyme function of a protein, okay, with 4 digits. So and the 4th digit is important because it's the substrate specificity, whereas the first digit is more like the enzyme class. So a machine learning method, every week you have a different method. And one of the things we did is we compiled this and see how good they were. And since the paper, there's been a lot of new methods that have come out. And I can show you graphs that shows that this method is really bad on novelty. But all these methods actually fail on anything that's new because it's not in the training set, okay? So by default, by definition, they fail. And where I think they were totally naive, and I think it's naivety, is that I'm going to show you that they could never have actually done what they claimed to do because it was impossible. The 500, like there's about 1,300 genes of unknown function in E. coli out of the 4,500. People have been working really hard for 30 years to try and figure out these, okay? If they had an EC number that was already known, that was already present, it would have already been linked to that gene. So the ones that remain are the ones where we really don't know. Mm-hmm. And what these guys were trying to do is link it with an existing EC number. So by definition, it cannot be correct. So pretty much you're forcing a peg in a hole, and it's never going to work. So by essence, there was a big flaw in that paper that none of the reviewers caught, that the editor didn't quote, because the field of functional annotation of protein is actually pretty small. People who really understand that is a few, and there's the biocurators, there's a few good bioinformatic guys, and there's a few experimentalists. So most experimentalists are very good in their area of expertise, and they're going to say, oh, there's a mistake in that annotation in that database, but they don't have a global view of how to annotate protein families. So there's the people who have developed the tools like the Babbitt and the Gert and me, who's really thinking about how to annotate protein families and separate paralogs correctly. We understand this. But I tell you, by fighting with the reviewers for a year, most people don't understand what a new function is. And also I discovered kind of a precipice between the ML guys and the experimentalists, because the function, the name unknown, has totally different meanings. So for the AI guys, you sequence a genome, you predict a protein, it's an amino acid sequence, glycine, lysine, it's unknown. You don't have a function. But what they mean, it's unlabeled, okay? So the difference is between a label. So all these tools, all the machine learning tools are actually labeling tools. And they're good, and their goal, actually, where we're going to use them the most is to propagate the known knowledge, okay? You capture all the known knowledge of publication of enzymes, and you try to propagate this to a new genome, and you're like, this well-characterized protein in E. coli, we label the same protein in this new genome we're sequencing. For an experimentalist, that's not an unknown. That's an unlabeled, and it's like you do a blast and you can label it, okay? For us, an unknown is some protein for no member of any member of the whole protein family this protein belongs to has ever been characterized. That's an unknown. So when you explain that to the ML guys, they're like, what? An unknown? How do you even start? And we're like, well, that's what biologists do. We discover the functions of unknowns for the last 100 years. Before it was through genetics and through biochemistry. Now we do it through data mining and bioinformatics, but we're still discovering the function of unknown. And what I do in my lab is really discover new EC numbers, new functions. So they cannot be discovered by AI because they're not in the training sets. So that I think is one of the reasons, is that this field is complicated. I tell you, because I teach it, it's complicated. Not many people are real expert. A lot of people are expert in sub bits of it, but very few people have the big picture. And if it was reviewed by ML guys, forget it. They just ate it up. But the editor is cool. The editor should have, like, I raised a flag, he should have taken the flag. He's not gonna like me, He thought I would go away. He
just made the paper stronger and more thorough.
Exactly.
Right. I mean, I can tell you, I tried to read the Kim et al. Paper first, and then I read some of it, and I read some of your paper. Yeah. And I think your paper did a really good job of explaining to me why your field is difficult, which is something that often bugs me when I'm reading scientific papers, is they don't really get into the nitty-gritty of like why something is hard. Yeah. And it's often just like way harder than anybody explains, because their paper is always fixing every problem that they highlight. So they only talk about the problems that they are then fixing. And then I'm like, okay, I guess it's not worth working on that anymore because they just solved it. Like, that's kind of the impression you get. But
it's also, I think that the Kim et al. Paper really followed what I found to be like a very standard format for a machine learning paper. And so it was kind of like, okay, this is the process you follow with machine learning, but it wasn't engaging deeply with the underlying domain, which is the kind of like other very, very crucial and foundational aspect. But it kind of like from a machine learning perspective, you're like, okay, this is the steps you follow. Although, like one of the things I really care about is that machine learning should be really engaging with the underlying domain very deeply.
Yeah, one thing that you raised, Valerie, was the idea of training data contamination in the paper. And when I was reading the Kim et al. Paper, I also didn't really get a good sense of how they tried, what they did to actually try to avoid that. Because if you just mix everything together, you're going to get every protein family in the training data and in the test data, and however you decide to label those different data sets. So what did they try to do to split this up, and why did that fail?
So I really tried to dig into their methods a lot to try and figure this out. So they used the 2018 version X of UniProt, and I went back to that version to get what were the annotation of the proteins in that version and to see where the data leakage could come from. That was very painful to do. So I think they just took the whole SwissProt, UniProt at that date. So there were the protein annotation of the protein family of the test, which the test is the 1,300 proteins of unknown function of E. coli. They didn't take them out of the training set. These were in UniProt, but with a vague annotation, or with the annotation of UniProt that was for that protein in 2018. I might have missed it, but it was not really detailed. But I don't think, from what I read, that they extracted the proteins of the test set from the training set. Out of the 450, 110, the annotation was already in UniProt. And sometimes the EC number was already there. Sometimes the EC number was not there, and they labeled with the EC number. We go back to this labeling thing. So I gave them the credit of labeling a protein that didn't have an EC number with its EC number. But I know a BLAST would do that, okay?
I do think the problem of constructing a thoughtful, well-designed test training split is often neglected, and it is like a totally crucial step. And I mentioned this in my blog post, but there's a paper I love called "Everyone Wants to Do the Model Work, Nobody Wants to Do the Data Work," and that it is a really common problem in machine learning, and that all the incentives are kind of set up around building a fancy model, but the really important foundational work of making sure your data set is well constructed often gets overlooked, and being very careful about how you do your test training split.
Yeah, I don't think that was done at all in this paper.
Yeah.
They trained on, as far as we can tell, everything in UniProt
at that date.
And so was
of the 2018.
And that was they were even, because I had wondered if it was that they were that sometimes the number or function was showing up in a different field of UniProt, but you think they just took everything?
They took the UniProt entry. If you take it, if it has an EC number, it'll be there. But a lot of the ones they had that I called already known, the annotation, the text of the annotation was UniProt, but UniProt had not linked it to an EC number. But for some they had, for some they had not. I don't remember the split. Yeah.
Why were some of those not already labeled if they were already found in the literature?
So you have to realize that UniProt funding is dismal. They have 2 people annotating the whole protein world. Okay. So this is the general state of biology today. We're generating tons of data. We're using databases on daily, minute. I use UniProt 200 times a day. Okay. I'm on UniProt all the time, but we're not funding really the databases. They have to scramble all the time to get their funding, because they're not funded. They're funded like us. They're funded like a research operation that have to do new things all the time. They do a grant where they should be foundational. So we're in the US, we're funding NCBI with tax money. UniProt is not funded by European tax money. They're funded, and actually NIH just cut they had a big subcontract from NIH that got cut because NIH just cut all foreign contracts. So they're scrambling to get funding right now. I don't know if UniProt will still exist when it's this foundation of most research we do in many, many labs. Wow. So the funding to capture these EC, so they're trying to automate as much as they can. They're trying to use machine learning to do that. Why I know all this stuff right now is because I've been trying to put the 3 communities that are involved in this annotation process are the experimentalist, the computational biologist, and the biocurators. And the biocurators are kind of forgotten. It's considered as, butterfly collection. It's not considered as science. And it's hurting all of us, because we're the foundation. When you do an enrichment, when you do an RNA-seq and you do an enrichment, you're based on gene ontologies and annotations that are put in there by curators. And when you talk to biologists, they have no clue how their stuff gets into databases. It's magic, okay? So there's not enough interaction between the 3. And I've been working for the last really 10 years trying to build this infrastructure, and I failed for the moment. We tried to do a center with NSF. We didn't get it. We tried to do an institute with NSF. It didn't even get reviewed with the destruction of NSF that's current. So I'm actually talking to a foundation right now. We might have a little edge there. We'll see. But there's no real place where the three communities interact. They interact 2 by 2, like biologists and computational and biocurators. But the three, my little meeting, I did a database paper a few years ago where I had put the first 3 together. It was great because people were realizing how the whole workflow of the data from the paper to the curator, and then so it's biologist, paper, so publishers, and then it's database, biocurators, and then the biologists use it again. And this workflow, we're all into it, but we're not really communicating.
Wow. Why do the biologists not go and suggest things for the database while they're publishing their paper?
Well, because there's no incentive. And we're actually, one of the things we were discussing in our meeting, in our brainstorming meeting, was if you publish a paper in biochemistry and you discover an enzyme function or you describe an enzyme, you have to do a little page describing your enzyme function so that UniProt can grab it automatically.
Mm.
But I can't tell you how many papers, especially chemists are fantastic for that, or biochemists, they publish an enzyme, they're going to do all the kinetic or discover its function, and they call it Robert 2, there's no UniProt, no NCBI, no identifier in the paper. You have to go in the SUP, get the oligos they use to clone, and blast those to figure out what the gene is. Like when you publish a PDB, a structure, you have to put it in PDB. It's imposed. When you publish RNA-seq, you have to put it in genome. There's no standard and constraint on how to publish functions. So it's the Wild West. Some people are aware of it, some are not. Sometimes I even get caught. We write a paper and I'm like, where are the UniProt accession numbers of the proteins we're working with? And so I go back and I make sure that we worked with this gene, UniProt accession X,
Mm-hmm.
But a lot of papers, there's no standard. And part of our centers were to try and do modules for grad programs to explain the whole like a biocreation module as an online module that we could give to grad programs in biology so they could understand how important it is to do when they publish to put identifiers.
Mm.
A gene name is not a unique identifier. And all my students fall into the trap. For example, you have a Y gene in B subtilis that has the same name of a Y gene in E. coli, but they're not the same protein families. So I'm like, when you do something and you find by your data mining, do a blast to make sure that objects that you think are connected are actually from the same family.
Wow. Yeah, I really hope that you succeed at some point in getting this stuff standardized.
Sometimes I'm very selfish. Sometimes I'm very selfish and I'm like, I know how to do this. It gets me 10 to 15 papers a year. I should just do it on my own. And, and sometimes I'm like, oh, but if everyone could do this and if the whole biology field could do it correctly, everything would go so more quickly and a grad student wouldn't spend 5 years on a wrong annotation and the gene ontology would be much more efficient. It's like, so I'm torn between the two. And, as older I get, I'm like, this is the last time I try. And, I don't know how long I'll have the energy to do this. Maybe Rachel will be in the generation that can take over if I fail. I'm trying to train also the new generation,
There's a paper I really like called Lessons from the Archives by UNCO Joe and Timnit Gabru that is kind of about all the things that machine learning could learn from the library sciences about how to kind of be more intentional in curating and organizing data, because I think machine learning has many of the problems that you're describing, Valerie, in the field of enzyme, where it can be quite haphazard with the data. And then also people are just kind of taking whatever data they can find to use, and it's not necessarily the most thoughtfully constructed or organized.
Right. And I feel like they will see something like UniProt. I mean, it looks very official. It looks like an amazing, complete resource. You wouldn't even think there are funding issues. You wouldn't even think that there are things to keep in mind while you're analyzing the data. You're like, this is the database. This is what I need. I need data. I have the data. And
it's also something with like, I think, so AlphaFold, is an amazing success in terms of having smashed all these records around protein structure prediction. And there are some really innovative algorithmic, new algorithmic techniques in it. So I don't want to take away from that, but it's also, I think people often forget, like the Protein Data Bank was set up in 1971. And so there have been people around the world contributing to Protein Data Bank for 50 years. And then the CASP competition started in the mid 90s. So you had for 25 years prior to AlphaFold, a lot of people really thoughtfully constructing this competition and thinking, how do you have, really making sure that the test set is completely held out from the training data that's available. And those things were very foundational for AlphaFold happening, to have this really, very large and well-constructed data set, and then also this really well-structured competition to spur on, the creation of these algorithms. And so that doesn't take away from the algorithmic innovations in AlphaFold, but it's like you need these necessary ingredients of kind of how you organize, the quality of your data, and then also how you organize it for training. And I feel like a lot of the kind of hype around AlphaFold loses that, and it's just like, oh, it's just this amazing algorithm, and we can do amazing algorithms for everything. And you really have to think, though, but okay, is the underlying data there of the right quality? And also, if you think of the work that the CASP competition did into, really having like the kind of like they were handling the training and test set, and I mean, people competing in it did have to make their own to kind of train along the way. But I think that really kind of helped with the standards there. And so I think often when people are just kind of, finding their own data set or going to UniProt, they're not necessarily, they may not even have the resources to put into kind of the construction and being really thoughtful about those data choices.
Yeah, I think that's such a good point. And what you were just talking about with AlphaFold and how there was a competition and everything, and you have the Protein Data Bank as like this amazing training set that they can start with. This is exactly why DeepVariant also worked as an application of AI in biology. I was on the DeepVariant team for 4 years, and before I even got there, there was a competition called Precision FDA, so it's done by the government, and they had actual training data effectively. The genome was already sequenced really well, but they had done another genome and they were holding that one out. And so exactly the same way, you have really high quality labeled data that people could use. And then there was a competition. And the competition was not meant to specifically identify like an AI method. It just so happened that that was what won the first time. And then while I was on the team later, there was actually a second like Precision FDA Truth Challenge version 2. And there, most of the methods were AI based. And that was just 4 years later. What's really great about these competitions is that they are holding out the test data so that nobody can accidentally train on it.
ImageNet and AlexNet is another great example of, what really kind of kicked off this deep learning revolution, or at least popularized it into the mainstream, of AlexNet winning the ImageNet competition in 2012. And again, that's something where, a team had been building the ImageNet data set for several years prior to that. And then, you have a very high profile winner that did, really introduced new algorithmic approaches and kind of smashed the competition, but you had a really kind of well-structured competition and data set to work with.
In the function field, there's the kappa competition that Ido Friedberg has been now leading for several years. And he's trying to do that. It was inspired by CASP, but to do it for enzyme function. And they struggled a lot to find ways to do this, test data, training data, challenge thing. And I think it did spur most of the good papers that come out on predicting EC numbers with machine learning comes out of people who were involved in that CAFA effort. But I think they're still kind of babies in the sense that they're giving points for figuring out correctly the first 2 or first 3 digit of the EC number, which for a biologist makes no sense. It's not sense, but it's not useful because it's really tough to get the 4th digit. And biology is about the 4th digit. So I think this competition is kind of helping improve tools and it's driving the field. But now is the time to really bring the biologist in so we can really ask the hard questions. They've been working on like kindergarten prediction type, Ido is not going to be happy. He's going to see this thing and he's not going to be happy. But
Yeah, and I would say like the biologist should have been included from the beginning, because I feel like that domain knowledge is it's so crucial. And this is true of any domain, that you really need to incorporate the domain experts for understanding kind of what Maria said earlier of like what makes this field hard and what are the kind of particular aspects of it.
So Rachel, partly why we came across Valerie's paper was your blog post, which kind of went viral on social media and on Hacker News. How did you come across Valerie's paper?
Oh, yes, actually, I took a course with Valerie and she mentioned the paper, and we actually didn't even cover it in the course, but I wrote it down since I'm into machine learning.
If you'd like to study with Dr. Valerie de Crécy-Lagarde, check out today's sponsor, the University of Florida's Department of Microbiology and Cell Science. They offer an online Master of Science program, and Valerie is one of their faculty members and teaches comparative microbial genomics. This online master's program stands out for a few reasons. First is the sheer number of courses, with over 40 graduate-level courses to allow you to customize your degree. And second, this program is entirely online, self-paced, and made for working professionals. Check out the link in the description to explore their program, review admissions requirements, and start your application.
But it really, this paper, I loved her paper, and it really captured a lot of issues that I've cared about for over a decade. Because really, I got interested in deep learning back in 2012, 2013, and could kind of see from the start, like, this is really powerful, but you're going to need domain experts to really understand the field. And in fact, such a kind of a problem just to take computer scientists that don't understand a domain and have them applying machine learning without that domain expertise. And so I spent many years trying to get domain experts from other fields to understand deep learning. Like, one of my theses was actually that it's better to take a domain expert and get them to understand and use deep learning for themselves than to have a computer scientist who doesn't understand a domain applying machine learning to that field. And then I also became very focused on potential risk and harms of machine learning. And a lot of those do arise from when people don't understand the underlying domain.
Were you surprised by the reception?
I was, yes. Something that was really neat was this really resonated even beyond biologists. I had a chemical engineer contact me and say, this is exactly what's happening in the search for heterogeneous catalyst. And I should say, I don't know anything about heterogeneous catalysts, but he told me that people are using AI to publish papers, and that if you actually know chemical engineering, often their results are either one, already known from the chemical engineering literature, and it's just like, oh, if you read the literature, this isn't a new result. Or two, people are saying that AI has discovered things that are actually thermodynamically impossible. And it's like, no, this wouldn't work as a heterogeneous catalyst if you knew the chemical engineering. And so I thought that was really neat, that I think this post, resonated beyond people that are interested in enzyme classification, but understanding like, hey, this is an issue that can show up other places as well if you apply machine learning without the expertise. And also that issue that Valerie was highlighting about the unknown unknowns, because that shows up in other fields as well, and that that's not what these machine learning approaches are designed to do.
So I want to build on the thermodynamically impossible idea. So a lot of the annotations that they came up with are biologically impossible. And it takes like a biologist. So for example, they discovered 12 mycothiol synthase in E. coli in the unknowns. It just takes me half a second to say they have to be wrong. First, you wouldn't have 12 of anything in E. coli, except if they have slightly different functions. Two, mycothiol is not E. coli molecule. It's found in mycobacteria. E. coli use a glutathione. And so then I talked to the machine learning guys and like, how'd you know it's wrong? I'm like, well, it's just biology knowledge of 30 years. But if you had you can combine this with a good metabolic model. And if you could do that in silico by taking your annotation and putting it in the metabolic model. And does it make sense in terms of, are the other genes of the pathway there? Yes or no? What's the function of this thing? Is there another thing that does the function? So what I do intuitively could be done bioinformatically if you put everything together. And one thing we emphasize in general in protein function is it's two-dimensional. You need the molecular function, which is the EC number, but you also need the context, which is the biological pathway. And you always have to make sure the two work together. Otherwise, you can have artifacts. So in vitro, you want to show that when you delete the gene in vivo, the pathway you think it's involved in is affected. Otherwise, your in vitro might be an artifact. And so, and it's the both other ways. So often in vivo phenotypes are very broad, and you have a broad phenotype, but you don't have the enzyme function, and you need that level. So when I work, what's really tough is to get both of these dimensions correct,
Coming from genomics, I'm struck by how unscalable this field sounds.
So this has been the issue that has been I've been working on for many years. And I think you were right until maybe 1 or 2 years ago. But I think now this whole institute grant made me learn to work with the AI agent guys. So pretty much what I was doing is doing storyboards where I take a gene and I explain everything I do. So this gene is of known function and I show it's not correct. All the operations I do to get to my conclusion. Or there's a gene of unknown function, but actually the function is in the literature but has not been captured. Everything I do to fix that. And then this gene is truly of unknown function. Everything I do to try and predict or a very specific function, or at least a metabolic area or something that can bring some knowledge to that gene. And I can write these storyboards, and now I have a way to show them. And the AI guys were like, oh my God, if we have these, we can actually build AI agents that do part of what you do. So what I do is very heterogeneous. I go all these databases and then I put in my brain. But I think now, if I can work, if people like me can work with the machine learning guys to kind of reproduce, because the problem is what I do is not scalable. How can we make it scalable? It's through using machine learning intelligently. And it's going to require a lot of work to get there. But I think at least now the tools are there. I've been saying, this is the way to go for 20 years, except there was no way to make it scalable. So I was kind of like in a void. But now I think it's exciting because we have the way to maybe make it scalable, or at least try to make it scalable, I would say. I don't know what Rochelle thinks about that. Yeah.
I perhaps have concerns about I worry that kind of more broadly in science that we may be under investing in kind of lab work and searching for mechanistic understanding. I think the emphasis on, and I love AI and data, but that I think often then there's this kind of emphasis on what's the easiest way to get the largest quantities of data approach, as opposed to really thinking about what's the right data for this problem, and what are mechanistic experiments or smaller experiments that are harder to do, but will yield kind of more interesting or more useful data. And so I hope that we can get the incentives right to still invest in those sorts of things.
Oh, in my vision, the machine learning part is to help the experimentalist do the right experiment.
Because you need that sounds great.
Yeah, you need that at the end, because some of these functions, I'm going to say, well, I can't say between this and that. We're going to have to go to the lab. Or I have this idea that this could be this function. It's totally new. We have to go to the lab. But I think there's first, a lot of the genomes are wrongly annotated because of the paralog over prediction issues. Because paralogs, they're part of the same family and they have subgroups. And you think that the function of subgroup A is the same as subgroup B. And it's not a trivial problem because 80% of the protein families are wrongly annotated because of that. And most of the genomes are wrongly annotated because of that. So I think that's what we're working on right now with Raquel, is to try and develop machine learning tools that can separate subgroups of families correctly. But I'm providing very highly curated families with all the subgroups curated and saying, this is function A, this is function B, this is, I don't know, but it's not A or B. And we're trying to see just by sequence embedding first, and adding structure embeddings, and adding gene neighborhood embeddings, and adding publication, can we do the work I do that is very effective, but very slow?
That's a fantastic vision. And I'd like to have you, sharing all these different types of data and staying very closely integrated with kind of how that's used in the model is, that's really great.
Yeah, this kind of also gets to one question that I had just reading your paper is, can you use AI to do, like if so many of the labels are easy, can you get AI to do it? And is there value in that above what you're already doing? Like you said, you could just use BLAST. Do you see value in the AI as well?
Yes, there is. I think the AI is getting better than BLAST in separating these paralogs. It's not perfect yet. So I would say, and what's clear is that the more difficult the protein family is, the more they fail. Like if you just have one amino acid that separates the substrate specificity, but this is where I'm really hopeful, because if you add structure into if you add the active site, if you can say, okay, this is the active site of the protein, and there's an amino acid that's critical for the function that's different in this subgroup. This is what we do manually. We go and find motifs that separate the subgroups. I think this could all be done automatically down the road with AI. I hope that I think it might take 5 years. I don't know. It goes so quickly that I don't know how long it's going to take. But I think the methodology and the thinking is there. To try and predict new functions that's a totally other story. But if we could get the labeling done right, that would be fantastic, because for the moment, it's not done right by BLAST.
Valerie, I'm curious to hear your take on, in the Kim et al. Paper, they do look for functional motifs and kind of offer that as like they're using attention to identify functional motifs, which I thought was a neat idea, but I'm curious, like how you feel about there.
I think this is the way to go. And for proteins so if you know the function of two groups, you have group A, group 2 that are very closely similar, and you can separate because they have this motif and the two groups, and you have label A and B, these tools are going to be able to better label the two subgroups of very closely related families. I do think that when you see all the methods that come since, other people have better integrated structure and things like that to do this better than the EC transformer tool, but I think it's the right direction. The problem is when you try and apply this to totally unknowns, where you're just trying to push an EC number. That's where they did their big, conceptual mistake in that paper.
Yeah.
I think, Valerie, what you were describing as like the ideal use of AI for you is what people would call a lab in the loop approach.
Yes, exactly. Yeah. User in the loop, expert in the loop. It has like different.
Right.
Yeah.
In this case, though, you would actually need to, like the AI would come back and say, okay, try to do this experiment next. How easy or fast is it for the lab to actually act on one of these? Because I think you have to do like an actual biochemical experiment every time, and you have to have the protein in the lab. So can you do this? Like if you have 1,000 proteins, like how quickly can you have these experiments happening in the lab?
So I've been doing that for 20 years because I discover functions. And if it's in the domain of expertise we master in my lab, which is the one I kind of focus on, we have the tools in the lab to test these functions. But sometimes I come up with a function that I don't have the tools. I reach out to the experts. I go to the expert labs and I say, you're the expert on this type of activity. And making protein now is not an issue, okay? We can make proteins most of the time, okay? So taking an enzyme and making a protein, and what's the issue is the actual enzyme assay or the analytical tool. If you take we do tRNA modification, we have to do LC-MS with standards. Like not anyone can just say, oh, I'm going to test this function, okay? You have to be in domain expertise. But I tell you from experience, when I email someone and say, oh, I have an idea that this gene could be doing the function you've been working on for 20 years and not finding, 10 seconds later, I have an email back saying, what is this gene? And then we collaborate, okay? So when you find the right person, so it would be it's not like one lab. So there were a lot of efforts of taking 1,000, 5,000 proteins and doing enzyme screens, but you were doing like lipase or, hydrolase, and you were taking standard compounds and substrates, which would never be the biological substrate. So you would be pretty much just saying, yeah, this thing can hydrolyze a compound, but that doesn't give you function. So to do function, you have to integrate all this data from the biological context, the gene neighborhoods, thinking about how like the distribution of this gene in nature, come up with some catalytic activity that usually is not standard, the substrate is not standard, or the activity is not standard, and then find the domain expertise guys that will help you. And for them, it's actually not a lot of work because they have the assay going in their labs, or they know how to do this. So it's not a one lab can do all enterprise.
That is really interesting and super difficult to study then.
So one of the ideas we had in our center was to include Adam Deutschbauer, who does a lot of TNSeq. And that's kind of a phenotype, high throughput way to look at function. And he's been very successful. So some of the phenotypes, some of the high throughput ways we wanted to do that was through genetics. But enzymology with a specific substrate is going to be tough to really automate on scale. So our way to kind of touch scale was through TNSeq. And his papers are great because they discover function because they have the phenotype. When you hit this gene, you become oxotrophic for this amino acid. And now you can go back and do a lot of comparative genomic on this protein and figure out which amino acid and whatever,
I think you're using some vocabulary that I am not sure I understand. Is this similar to a knockout?
So it's similar to a knockout in the sense that you take a population of bacteria and you saturate them with transposon that knock out pretty much every position of the whole genome. You pull them together and then you grow them under a selective pressure. It can be temperature, it can be no amino acid, it can be a carbon source that they have to use. And then you let them grow, and it's just a few days, actually. It's very quick. And the mutants that have a hit, the cells that have a hit in the gene that are necessary to grow under that condition are going to be eliminated from the population. So you sequence again at the end, and you see which genes are still there and which mutants are still there and which genes are not. And you give a TNSeq score, a fitness score, pretty much. And then they have a database that's amazing. All my biology friends are on it all the time, and it's a very powerful tool.
So instead of starting from knowing what the gene is and looking for the function, they pick a function and then they pick
a phenotype. They pick a phenotype growing on this weird carbon source, using this polyamine as a nitrogen source. And then you can figure out all the genes of the pathway that are involved in that. And then you have the genes, and now you try and but it just gives a phenotype, no growth on polyamine. But then to find the EC numbers, you require a lot of bioinformatic and comparative genomic and trying to figure out these genes, why would they be involved in polyamine growth?
That's really cool. I love that idea of working backwards.
Yeah.
Yeah.
It's very, very powerful. You should read his papers. Yeah.
This is great. I just on a personal note, I've spent so long in genomics, and it's just really cool to become more familiar with your field of like figuring out enzyme functions.
When I try and sell my thing, I'm like, every RNA-seq, every metabolomics, like everything you guys do is relying on understanding the function of the genomes you're interested in. And if these are bad, what you're doing is or bad or partial. You're only capturing a little bit of what you could be capturing if the genomes were better annotated. And that's where, as a whole field of biology, we've been failing because it's a hard problem and because it's not exciting to curate and annotate. Okay, so funding agencies have, and I've talked to program officers like so many times, and it's kind of dichotomic. So when I talk to them one by one, I convince them. You can tell that I'm pretty, I can convince people. But you go back to the hydra of the big, NSF or NIH, and then I get two feedback back. I get, it's trivial, AI can do it, machine learning can do it, whatever. Why even pay people when computers can do it? That's one view. And we're not going to discuss how wrong that is in this, because we've just been doing that for 30 minutes. The other view is that it's so hard that we would just use the whole NIH or NSF budget trying to do it. And I'm like, maybe there's a middle ground between the two that we could find to move the field forward without just chaos, because now it's just chaos,
Yeah, so how would you explain what the point is? Like, if the functions have not been discovered by now, just playing devil's advocate, are they even important? What
No.
what
So
does it matter if we curate everything and we give them all EC numbers and it's all perfect? Like, where does that get us in terms of something that would help, the average person?
So you're never gonna get 100% of every genome. That's not gonna happen. Okay. However, what I'm saying is, if we already got the ones we knew already correctly propagated along the genome sequences we have, that would be foundational, because for the moment, even the knowledge we know is so labels are badly propagated. Okay. Most of the annotations we have today are not experimental. They're computational. Like the experiments have been done on like 0.01% of the proteins in UniProt. Most of the annotations in UniProt are computational. And that's not going to change. We're not going to do the experiments on millions and millions of proteins. So we have to make sure that the computational predictions are correct. And for the moment, they might be correct in a few model organisms where there's been all this biocreation effort. But as soon as you get away from that, they're wrong. But big wrong. Like, you even take, I've been we looked at Pseudomonas putida, which is a workhorse of bioengineering. People use it a lot. I would say half of the genes are wrongly annotated. And this is a bacteria that's really well studied. If you go to Myxococcus xanthus, 70% of the genes are known. So how can you think you're doing systems biology when you know a third of the components? So when you do any system biology, RNA-seq, you're seeing the genes that are up and down or co-regulated. Their function might be wrong, or their function might be unknown, when it should be known and correct. The data is there. It's not new data together. It's just high quality propagation, okay? Then there's bacteria are this whole metabolic, incredibly creative, like there's a lot of enzymes to discover in bacteria, and they would be pretty easy to figure out if we put the effort in using TNSeq or bioinformatics or both. Like there's a lot of, so we could bring maybe the state of most bacteria from 30 to 50%. If we could bring it to 50 to 70%, that would be already improving the quality of what we do as a community 2 or 3 times. Then there's the 30% of the very hard cases, okay? The ones that we don't know the function, the ones I work on, okay? So the ones I focus on are the ones where I get a lot of bang for the buck. They're conserved from E. coli to human. So when you find a function of a gene that's conserved everywhere, then you annotate millions of genes. Then, of course, if you're an expert of like a weird bacteria that degrades toluene in somewhere, and you're the only one in the world interested by it, you might focus on the 10% of the genome that is involved in toluene degradation only in that organism, because it's your field. And if you combine all that, little by little, will improve the whole knowledge, maybe to 80, 90% one day. 100% is impossible. I don't think we'll get there.
So let's say we do improve that knowledge. Do we use it for medicine, agriculture?
Yes, everything. Everything you do now is based on a genome sequence to start. Like everything, like pretty much every biology project starts with a genome sequence or with an RNA-seq experiment. Or like you use genomic data now pretty much in 80% of biology. But the foundational of the annotation of this genomic data is bad. So making it better will influence everything.
I guess I'm looking if you could draw an example of where this has happened. Perhaps someone found a new function in E. coli and they were able to use it.
So in my work is not to discover disease. I'm a very fundamental scientist working in E. coli on tRNA modification. So I've discovered about 40 tRNA modification genes. And then I publish the paper, and then I get contacted by a pediatrician who can now understand why their patients have the molecular causes of the patients, because now we know the function. They knew that their mutation mapped to a gene. They had that information because they had sequenced the baby's genome, but they didn't know what that function was. Okay, wow. I come in, and this gene is conserved between E. coli and human, and I can say, well, now this gene does this. So sometimes it doesn't lead to a cure, but an understanding of the disease. Sometimes, so there's one family that we work on that's conserved from E. coli to human that's involved in a vitamin homeostasis, vitamin B6. And in this case, when you give B6 to the patients, you can cure them. And we're trying to figure out, I've been working on this family for 20 years. I know it's related to PLP, pyridoxal phosphate, and B6 metabolism. I still don't know the molecular function of it, okay? So sometimes it takes 20 years, 30 years to nail the two dimensions. But I'm not doing it for that, but because these proteins are conserved, people are kind of mapping knowledge, and then I realize that it's important for this disease, or it could be a drug target. There's a few protein families that can be antibacterial targets that have been developed because we know the function now, and we can kind of target it with small molecules. And we know this function is not in human, but only in bacteria. So we can, and we know it's essential. So but then this is because of my specific work. But then anytime you do an omics experiment, you're relying on functional annotations. So the quality of your work will depend on the quality of the annotations. If you do an RNA-seq of plants, how they react to stress, and you see a pathway coming up, but none of the genes are correctly annotated, you might, or they're wrongly annotated, which is most of the case, you might think that this is involved, like you might go into a wrong path because of the annotations are wrong.
I mean, one of the things I find interesting about this whole thing is that, I mean, you've spent a ton of time just digging into this one paper, trying to reproduce their methods, trying to find the problems they ran into. There's this quote that says that it's an order of magnitude easier to make BS than it is to refute it. And like,
tell me about it. Yeah, tell me.
Now it's a lot more than an order of magnitude because you can get the AI to make BS, but you need a human to refute it.
Right. And like, how do we stop this from like continuing to waste your time? I guess, you know, if they hadn't done it, someone else would have done it. So it's, I think what you did was really useful so that people are aware. So they have this thing in their head that goes, wait a second, okay, I've heard of this before. Maybe I should be more careful.
I think it's with any new field. You have this like explosion, and then you have things that people realize should be done differently. And I think Rachel is really thinking about that a lot. She can talk about this. But I think every new field in biology goes through this hype and down cycle, and then goes back to more like thorough. And I hope that we're in the same type of things. But Rachel should have some thoughts on this.
I mean, there's a few things that possibly make it worse. Are one, automation bias, in which people are more likely to think that computers are 100% accurate and don't make mistakes. And there have been some pretty disturbing examples of, you know, like local police departments buying like AI software and being like, oh, the AI is 100% accurate because computers don't make mistakes, and so we don't have to worry about this. And that's, you know, we all know computers, of course, do make mistakes, and they do what we program them to do. And, you know, it's not, but those biases are a concern, I think, with AI. And you see a lot of kind of black and white thinking of people either being like, this is amazing and it's going to eliminate all jobs, you know, or this kind of like extreme, you know, against it, not recognizing like, hey, actually, this is useful. So that's a concern. And then I think also kind of what Robert brought up about, well, the scale of refuting false information, but also just that really computers are designed to scale. And that's a lot of what the some of the risk and harms come from when you really can do things at scale. And so you're not just making a mistake, you can make a mistake at scale and you can make a million mistakes. And so those are significant challenges. And I think they often overlap with the low cost. And so, algorithms are, you can do something at scale very cheaply, and perhaps more cheaply than hiring the biologist to be in the lab testing what the enzymes actually do. And so, these are all obstacles we'll face. But I hope that we can counteract some of the hype to address these.
But this has always I had a student write a whole paper because there was an annotation based on bad experiments that got captured by UniProt, propagated like wildfire, and was totally wrong. And it was easy to refute just based on metabolic logic, because, for example, it was said to be a folate gene, and it was in genomes that didn't make folate. So when you have that annotation, you know it's wrong. But because there's not the safeguard, like UniProt does not do biological context. And I talked to Alan Bridge, he's the head of UniProt, and he's really amazing. And so because you can propagate without checking against biological context, you can have these type of mistakes. And it took a grad student 2 years to write a paper to refute it, So it's not just AI that does mistakes and has to be refuted.
Yes, yeah. And I'll say a positive is I thought it was really great when so NeurIPS is one of the major AI academic conferences, although also with a large industry presence, and there was a lot of discussion about how algorithmic and model work was more rewarded and more incentivized than data set work. And so they introduced an entire track on data sets and benchmarks to really try to incentivize and offer, opportunities for publishing and speaking specifically on data set and benchmarks. And so I think a positive is that there has been more recognition in the machine learning community of hey, we actually need to incentivize and recognize this work on kind of the quality of data sets and benchmarks, and not just the kind of the algorithmic innovations.
And one of the things we're doing, Raquel, and I wouldn't have to thought about this had not done all this work with, is the sequence similarity networks we're producing that are highly curated. I think they would be a great benchmark for any EC number prediction.
Yeah.
So I think we're going to try and make that as so it's going to take a lot of work to make it a data set that AI guys can use and to benchmark their stuff. But I think it's worth the effort.
Oh,
and what
great.
you're saying is kind of pushing me that direction. Yes.
Can you use AI to help you clean it up to make it ready for another AI to use?
I don't know. I haven't thought about that yet. We're just in the beginning, yeah.
Since we're on the topic of hype, recently there was a 60 Minutes interview where the DeepMind CEO said that, he couldn't see why not we could cure all diseases in 10 years or so. On a scale from 0 to bonkers, where do you see that statement?
I don't think you can cure every disease in 10 years with AI. I don't think that's going to help. Yeah.
Right, but it's not going to be even just AI that does it, because you need to do stuff in the lab.
There's so many diseases where we're really missing a lot of mechanistic information, and kind of talked about these concerns earlier, but that data that is more invasive or time intensive to capture is often not captured. And so an example I think now is that you'll see a lot of medical analyses, maybe using EHR data, which it can be interesting, but they miss a lot. There's so much that's not recorded in electronic health records. There's also a lot of racial and gender biases that go into how that data is recorded. And you have to think about all the illnesses that we don't have easy or clear biomarkers for yet. And I do worry that kind of a lot of, in general, incentives are around let's just use whatever data we have without thinking is there, additional data that we need and would be, offer deeper insights, even though it's going to be harder to gather.
So he did get a Nobel Prize recently, and usually Nobel Prize people wait, a couple years before making crazy statements.
Although, I think tech CEOs are kind of a separate category. So, and I'm not just talking about him in particular, but you do have a lot of tech CEOs that have made big pronouncements, and to keep in mind that they do have an area of specialty, but they don't necessarily have deep expertise in terms of understanding, policy and sociology and anthropology and history and kind of all these factors that are going to influence kind of how medical AI develops and how it's deployed and what problems it can be accurately applied to.
So one thing that I was concerned about is that on some of the social media platforms I saw in the discussions, there were people trying to defend his position, which is kind of slow down progress, probably, because of people thinking, well, we don't need to do this because AI is going to take care of it, or we don't have to invest in this.
But this has been the same in bioinformatics. For the protein annotation problem, people were telling me we don't need biocreators, we don't need experimentalists because bioinformatics is going to do this. So this is something we're facing between the need of high quality experimental data and the ease of computational analysis, as Rachel was kind of emphasizing.
It's interesting to kind of compare the fields, because I do think I'm working in the very baseline of genomics. Like if you can just read the entire haplotypes, like the 2 haplotypes of a person from end to end with sequencing and have it be 100% correct, then I'm out of a job. Like every type of research I've ever done is just to get that sequence right, to call the variants correctly. And that has gotten so much easier just with the technology, just the sequencing technologies. The reads are getting longer, they're getting more accurate. And that alone has just made such a huge difference in like the short time I've been in my field, which is about 10 years.
So imagine me, I was sequencing by hand during my PhD with Sanger sequencing. So science goes fast. Yeah.
But it's interesting that some areas like this can go really, really far ahead, right? So now the genomics has become kind of the trivial part. Like getting the DNA sequence correct is now the easy part, but there's still other pieces left, as I'm learning about from reading your paper, that are actually still really difficult. And it's just it's interesting to me to think about how can we scale it? Like, what do these frontiers continue to be that are the most difficult?
Well, it's also linked to funding. We have to go back to funding. There's not been an effort in that funding area. There's been dabbling, but because it was so hard, it kind of fizzled. And so, it's also about timing. People who are developing sequencing 30 years ago were like, we took a whole 5 years to sequence one genome, and now it's done in an hour. So I think there's also the timing, and maybe now for this field, the timing is right to scale.
I would also say medicine involves so many different factors. Yes, there are plenty of diseases that have a genetic component, but there are also, the environmental exposures, the infection, triggers by infectious agents, microbiome differences, then you've kind of got like multiomics with the, transcriptomics, like once expressed, and even seeing people who experience the same environment developing different diseases. But I think that a lot of I think sometimes people look at progress in genomics and are like, oh, medicine's almost solved. And it's like, wow, there are like so many different layers and factors that go into various health issues. And I think these, statements of saying we can cure all disease really are based on a very oversimplified view of what causes disease.
Knowing what you both know now, how do you separate hype from reality when you're reading a paper? How can you evaluate a new AI paper that, is proposing some kind of method in biology? What should you look out for? What should our audience look out for if they're reading a paper in their field and trying to evaluate whether it's complete BS or if it's something to be taken seriously?
I think it's a difficult issue when you're not in your field, because for me, it's very easy to do when it's in my field. I can see the predictions I make and I know immediately if they're good or bad. But if it's not in my field, I'm kind of a newbie and I have to kind of trust. So we have to go back to better quality publications and better reviewers, because it's going to be tough. It's easy in your field. It's tough not when it's not in your field for me to do. Yeah.
Yeah, and I think that really highlights just how crucial domain expertise is, and to make sure, that we're keeping domain experts in the loop to be reviewing and involved with papers. But it is something where you do really need to understand. I do more broadly think that everyone in society is going to need some level of AI literacy, just for recognizing, there's just so many false claims being made. And so and some of them, it just really even recognizing what AI can and cannot do, and really thinking about okay, what training data would even be available, and is it even possible that this product can do whatever is being claimed? But it is an issue that there are a lot of people kind of making misleading claims, often to sell products, or in some cases, kind of within their research.
I think on the topic of how to catch this in the peer review process at a journal, I think we need reviewers not to be more selected as experts in the problem that's being solved with the AI. If they send this paper only to people who know about AI, but not people who know about enzyme function prediction, then they're not going to be able to catch exactly the issues that Valerie saw very, very quickly when you read the paper. And similarly, I remember having been asked by a journal to review a paper because it contained visualizations. It was a visualization web portal thing. They made a web app. And I've made web apps, but I make web apps from my field of expertise. And they were doing something completely different. I should not be reviewing a web app for an area of biology that I'm not an expert in, because the point of the paper is not we made a web app. And similarly, we used AI should not be the main feature that the peer reviewer should be reviewing. Although all of us having some understanding of where AI can fall down, such as training data contamination, is important. But I think the biological expertise to evaluate the claims they're making is even more important than that.
Yeah, and then something we discussed earlier that I think could be useful is this notion of competitions, of just kind of when you have a team of people who are involved with curating the data and doing the test training split. I think that is offering kind of this additional layer of safety, that this has been well designed. Because in some ways I do feel maybe it is I don't think people should publish poorly designed papers, but in a sense, maybe it's a lot to ask people to really do a very thoughtful data curation and kind of all those steps just along the way, to doing a one-off paper. And maybe that is something that we need to be doing kind of more collectively, of developing okay, what are these kind of canonical data sets we want to work on for given problems, and then making sure we're really thoughtful about how we're doing training test split.
So I told you about Adam Deichbayer, who does this TNSeq. They've published a kind of a data set of enzymes that they discovered that are not in UniProt, and they discovered the function using TNSeq. It's called the price data set, and now AI guys are using it a lot to test their EC number, their function prediction tools. So it's pretty much every paper has the price set now in it, and it's great because it's a tough one. And there's a paper that just came out last week who separated the cases function of easy, medium, and hard, and you really see the more difficult cases. Most AI tools machine learning tools cannot predict them. So if you take an easy data set, you have 90% accuracy, but that's not that. You don't need AI to do those. That's the problem. So, so to have the good data set is very useful, as Rachel was telling.
Yeah, I think especially these days with AI existing, having these sorts of highly curated data sets is super important. And I want to shout out in my field, Genome in a Bottle, which created these variant call sets that are so important. And like DeepVariant could not exist without that Genome in a Bottle data set. That was then also used for that Precision FDA challenge. So it's really just kind of so important to generate these high quality data sets. And I love that idea of also with that price data set you mentioned, Valerie, that they separated it out and kind of, picked exactly 100 proteins, I think.
140, yes, yeah.
100,
yeah. So I think the problem is there's really no funding for biologists to do that. Because we can't apply for AI grants because we're not AI person. So unless they start doing grants specifically to generate data sets for AI that are funded, I don't know who will fund them, the experimental or the AI guys, there's no space. And I can't put that in my experimental grant, really, because I'm going to say, why? You're going to spend like 2 years curating something that you're not even going to use. So there's the funding for these type of effort is still not really there.
Yeah, and I think that's a huge problem, that I think AI has kind of hit a lot of the low-hanging fruit of what data sets were kind of like already available and curated, and that, we really need the continued funding and just also the continued funding for all these other domains, and recognizing like this other domain expertise is super important and crucial, whereas with the hype, the AI is kind of getting disproportionate funding.
I'm not sure how to tie this in with the rest of the conversation, but Rachel, you already have a PhD and you're doing another one.
Why?
Yeah, no, this I don't even necessarily recommend this. I did spend quite a while working in between. I worked in the tech industry as a software engineer. I co-founded Fast AI to make deep learning more accessible. I worked at the University of San Francisco, and I started a Center for Applied Data Ethics. You don't need, depending on what you want to do, you may not need a PhD. I think I really try to just follow my interest and my curiosity, as well as finding people that I'm aligned with on values to work with. And so, this was not my plan at all. But a few years ago, I just got so fascinated with immunology and microbiology. And at first it was like, I'll just take like a few online classes. And then I was like, I kind of exhausted my options, and was like, okay, I guess I'll get a master's. And then as part of my master's, I met a few professors I really connected quite well with. And they were like, have you thought about doing a second PhD? And I was like, and it seemed like I, did want to get to the cutting edge of microbiology research and better understand, like, what does it take to write a microbiology paper? So I've kept going with it. But I think with my math PhD, which was quite a long time ago, I did that straight from undergrad, and I don't think I had a good enough sense of actually what the opportunity cost of doing a PhD was. I wasn't really thinking like, hey, I actually could have learned a lot by working in the tech industry during that time instead. And also probably less of a sense of what I'm interested in than I do now. So I think I am bringing kind of more background of like, okay, I have spent a lot of time working and exploring different things, and have more of a sense of what I'm interested in, and kind of who his values aligned with me. But it happened organically, and it's not the plan.
Wow. So this is the PhD that you're like really interested in.
Yeah, and I should say, like, I loved math when I was younger. Like, it wasn't, like I was quite passionate about math, and I wanted to be a math professor. But I didn't have like probably a good enough sense of like, there are a lot of other things I could do with math, and kind of in the direction I ended up going in tech. I don't know that I needed the math PhD.
Awesome.
But now it's just like, there's still more microbiology for me to learn. And this really grew out of, for years I've been teaching my students that, like, if you're going to do deep learning, you really need to understand the underlying domain. And so I feel like I'm taking my own advice now, and I was like, okay, like I want to be working on these problems in microbiology. And so I want to make sure that I really, really understand the underlying domain thoroughly.
I picked biology as a college major back in the day. I basically was just choosing between the sciences that were available at the college I chose. So it was biology, chemistry, and physics. And I thought biology seemed to have the most unsolved problems that we would never run out of things to study, because we'll always find another organism and apparently another protein, even in the organisms that we already know well. I didn't know just how unexplored even our most understood organisms are, like E. coli and human, still have genes that we don't know the function of. That's incredible to me. And yeah, I guess I made the right choice, even if it was for relatively naive reasons.
Yeah, for me, what really captured my interest in the last few years was kind of learning about all these kind of surprising long-term health consequences. So like the research on Epstein-Barr virus causing multiple sclerosis was just like kind of mind-blowing to me. And, that's something where there is also a genetic component and, there are other aspects, but like, wow. And then also everything that's coming out about, potential links between like herpes viruses and Alzheimer's and neurodegenerative disease. And then I've been studying type 1 diabetes a lot, and that can be triggered by an enterovirus. Those were really, really interesting to me. And then as I dug in, being like, wait, all these also have this genetic component, and they have these microbiome differences, and feeling like, oh, and I spent like 20 years building skills in data science. And now I've got these problems with these like multimodal data sets that are really fascinating. And so I do really, want to bring my background in data science and machine learning to these sorts of problems.
Yeah, we will never run out of those problems to solve, probably, but I think AI and data science and so on should speed us up at least. It's at least helped with some of the fields that it's been applied to so far. So hopefully it'll continue to become useful, and hopefully we won't waste our time with AI slop and having to refute all of it. Because it can very easily go in that direction, as we have seen with just LLMs in general culture outside of science. Hopefully we don't see the same thing happening in science, although we are already seeing it clearly. So.
Well, on that positive note,
well, we can have a more positive note. Do either of you? We should probably just end on Rachel's positive note.
Yeah. All right. Well, thank you both so much for coming on. This was very fun.
Yeah, we learned a lot, and it was actually really interesting. So yeah, thank you.
Thank you. It was fun, indeed.
Thank you.